Hello, and welcome to this Nature.com custom webcast sponsored by Thermo Scientific. My name is Jayshen Carbon, and I will be your moderator for today's webcast, Biomarker Discovery, Translating Proteomics into Clinical Diagnostics. Thermo Fisher Scientific is a world leader in serving science. Its four premier brands, Thermo Scientific, Life Technologies, Fisher Scientific, and Unity Lab Services, offer a combination of innovative technologies, purchasing convenience, and comprehensive support. Thermo Fisher strives to help its customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. Today I have the great pleasure of welcoming two speakers. The first is Eleftherios Diamandis, who currently serves as Division Head of Clinical Biochemistry at Mount Sinai Hospital and Biochemist-in-Chief at the University Health Network, as well as the Professor and Head of Clinical Biochemistry at the University of Toronto, Canada. The second speaker is Andrei Jabovich, who is currently a Senior Postdoctoral Fellow at the Lindenfeld-Tannenbaum Research Institute at Mount Sinai Hospital and University of Toronto, Canada. Before we begin, a few brief announcements. After the presentation today, the remainder of the hour will be available for a live question and answer session with you, the audience. So ask the speakers a question, please type it in the questions panel at any point during the presentation and we will answer them today. We will try our best to answer as many questions as we can, and we'll follow up with those we didn't have time for after the session. Finally, a recording of this webcast, including the slide presentations, will be available to view again on demand on our website, nature.com/webcasts. If you're watching on demand, please feel free to send in your questions up to a week after the live broadcast, and we will answer them via email. And now, over to Eleftherios.
My name is Eleftherios Diamandis, and I work at Mount Sinai Hospital as a Biochemist-in-Chief. I also have a very active research laboratory which is interested to apply genomics and proteomics technologies for early diagnosis of human diseases, especially cancer. In this webinar, we will try and explore the issue of applying proteomics in translational research, and especially for developing new diagnostic tests for cancer, infertility, and other diseases. Proteomics is a technology that was developed approximately 20 years ago, and everybody thought that proteomics will revolutionize clinical diagnostics. However, as time went by, it has been demonstrated that there are many difficulties associated with the application of proteomics in diagnostics. And one of the hard realities in this field is that objectively very few, if any, clinically useful biomarkers have been discovered by proteomics technologies over the last 15 years. And although some people claim that this is due to the lack of funding, this is actually not the case, because there is enough funding in this area of investigation from both granting agencies as well as commercial organizations. The expectations for this technology were very high over the last 10 years because the human genome has been delineated, and it was thought that with the whole knowledge of human genome and all the genes that are encoded, it would be possible to quickly develop diagnostics based on genomics and proteomics technologies. There have been, the last 10 years, spectacular advances in mass spectrometry, and these advances have enabled people to characterize whole proteomes, and this is why people expected that these technologies can revolutionize diagnostics. However, it has been realized as well that there are many major obstacles with clinical proteomics. These include the lack of standardization of workflows and irreproducibility of results between laboratories. Other people thought that the appropriate biospecimens have not been used, and many of us are using the so-called convenience biospecimens, which is biospecimens that we have in our freezers. But these may not be the best ones to do a high-quality discovery. As I mentioned before, there was also very high expectation that the new biomarkers will be much better than the old ones, the so-called super biomarkers, which supposedly could have almost perfect sensitivity and specificity. However, we need to realize here that such super biomarkers may actually not exist. The literature has also been plagued with something that we call fast discoveries, that is, discoveries which are published sometimes in very prestigious journals, but they turn out to be not true in subsequent validation studies. On the other hand, proteomics has some major advantages over other technologies. For example, clinical proteomics are not in a need for availability of high-quality antibodies, which are the workhorse of the proteomic measurements until now. Proteomics is also inherently a multiparametric technique, which means that we can measure multiple analytes at the same time and develop panels of biomarkers. Mass spectrometry can also be a very highly specific technique based on the selected reaction monitoring analysis, which we will describe a little bit later. Also, mass spectrometry has this inherent amazing ability to detect, quantify fragments and isoforms of proteins, something that is not usually possible with antibodies. And mass spectrometry can also detect post-translational modifications, which again is very difficult to do with ELISA technologies. Now, one major point that I want to raise here, and people are looking for the answer, is if proteomics can actually replace ELISA. And I say here with confidence that proteomics and ELISA are actually two complementary techniques. It's not that one will eliminate the other. It is also interesting to examine if proteomics has any major notable achievements over the last 10 years, and I can mention two of them to facilitate the discussion. One is that the so-called MALDI-TOF technology has now been used very successfully for identification of microbial infections in a very quick manner and very accurately. And this technology has now been cleared by the Food and Drug Administration for routine use in microbiology laboratories. Another area of research for which mass spectrometry has enjoyed tremendous success is in the typing of amyloidosis, a disease that is associated with aggregates, and now mass spectrometry can delineate the composition of the aggregates for much more accurate diagnosis and much more better selection of therapy. I think we should examine also what are the future trends with mass spectrometry. My belief is that over the next few years we will see better instrumentation that is more sensitive and is faster than the current mass spectrometers. I think we will see better software with which we can do data reduction much faster and more accurately. I think sample preparation techniques will evolve, and these are necessary because in the clinical laboratory one would need some kind of automation for processing a large number of samples. I also believe that there will be evolution of a technology called immuno-MS, which combines the abilities of antibodies with mass spectrometry for enrichment of the analyte of interest, followed by quantification by mass spectrometry. I also believe that the mass spectrometry, with its inherent ability to detect mutated proteins with single mutations through the selected reaction monitoring analysis, will likely revolutionize the quantification of true tumor markers that we can only see in cancer in biospecimens. In this slide, I graphically describe why there are difficulties with discovering and introducing biomarkers into the clinic. One very minuscule reason for biomarker failures is fraud. And although fraud has been identified in a few high-level publications, it is not a major cause of biomarker failures. I believe that one of the major causes of biomarker failures is so-called the true discovery, but with weak clinical performance, which means that we can discover a lot of biomarkers with mass spectrometry which are characterized by low specificity, low sensitivity, and low positive predictive values, thus providing information which is not very useful for clinical decision making. Another major cause of biomarker discovery failures is the so-called fast discovery, which, as I mentioned before, refers to publications, sometimes very high-impact journals, which cannot survive validation in subsequent studies. And it has been identified that many of these fast discoveries are due to either pre-analytical, analytical, post-analytical, and bioinformatic artifacts.
Thanks, Eleftherios. And now over to Andrei.
My name is Andrei Drabovich, and I work at Mount Sinai Hospital, Toronto. I'm actually actively involved in biomarker discovery, and I use proteomics and mass spectrometry to discover, identify, and verify novel biomarkers of several diseases, including cancer. So in the following slides, I will present our approach to protein biomarker discovery by mass spectrometry, and also present some specific examples such as identification and verification of biomarkers of some urological diseases. As an introduction, let's consider different types of molecules that can be actually used to develop molecular diagnostics. These can be gene mutations, SNPs, mRNAs, lipids, metabolites, and proteins. These molecules have different advantages and limitations when it comes to developing molecular diagnostics. For example, gene mutations and SNPs can be great to predict disease predisposition, but may not be useful to diagnose the acute phase of the disease. If you look at the actual number of molecules and diversity, there are around 20,000 human coding proteins and genes, and these genes account for around 50,000 transcripts and around 40,000 metabolites, and all this diversity accounts for as many as 1 million different proteoforms if we consider SNPs, alternative splicing forms, and post-translational modifications. So this diversity of proteins and protein isoforms, and also their secretion into biological fluids, makes proteins as a very promising candidates to develop new molecular diagnostics. If you look at the potential use of protein biomarkers in the clinic, this may include disease screening, diagnosis, prognosis, disease staging, monitoring effectiveness of therapy, disease and disease relapse, and predicting therapeutic response. If you look at the actual number of FDA-approved markers, we can find quite a few blood and tissue-based markers for therapeutic response and disease relapse, only some markers for prognosis and disease staging, and there will be a very limited number of markers for diagnosis, and a very limited number, or maybe even none, for screening. And there is some controversy about the use of the prostate-specific antigen as a screening biomarker for prostate cancer. The lack of screening biomarkers may be explained by the challenge of the issue of finding and identifying an ideal screening biomarker. Such biomarkers should have very high positive predictive value and also extremely high sensitivities and specificities. Its tests should be inexpensive and non-invasive, and markers should provide sufficient lead time, basically the time between asymptomatic disease and metastasis. So now if you look at the performance of some markers in the clinic, for example PSA, we can see that positive predictive value of PSA, for example, is only 30%, which means that 70% of men who are diagnosed with PSA go for biopsy without having aggressive cancer. And if you look at some rare cancers, such as ovarian cancer, then we realize that ideal screen biomarker for this type of cancer will require markers with almost perfect sensitivity and specificity, which means that this problem is really challenging, and for some rare diseases they may not actually be feasible to develop great biomarkers, great screening biomarkers. So the challenge of developing new biomarkers for early diagnosis and also failure of some markers to reach the clinic lead us to the conclusion that we may seriously reconsider some of the criteria which are used to select biomarker candidates. And in our work, we came up with a set of criteria that we actively use in our work. For example, we focus on secreted and membrane shed proteins, proteins found in proximal fluids, tissue-specific proteins, and proteins involved in pathways related to disease progression. We think that, for example, there may be very low chance that nuclear or intracellular protein will emerge as a very specific biomarker to diagnose cancer or other diseases in biological fluids. And then if we want to increase specificity of test, tissue-specific proteins may be one of the solutions to increase the assay specificity and diagnose very specific certain disease. In our work, we use several techniques and approaches. Mass spectrometry-based proteomics is a core technology in our work, and we also use several integrated approaches such as analysis of proteins in proximal fluids, cancer cell lines, tumor tissues, some animal models. We also use some microarray profiling data, and finally we analyze all these proteins in blood, usually by ELISA-based assays. We also have capabilities for protein expression and antibody production, and also ELISA development, and we are involved in several multi-center collaborations to validate our new novel biomarkers. In our search for new biomarkers, we follow a typical biomarker discovery pipeline which consists of several well-defined phases. It's an identification phase, verification phase, clinical validation, and the final goal is to have an FDA-approved clinical assay. There is an increasing number of samples when we move down the pipeline, and a decreasing number of analytes or proteins at each step. So first step of biomarker discovery, identification step, is very different from the rest of the phases in the pipeline. For the identification, we use standard shotgun bottom-up proteomic approaches. This is a slow step, it's not high throughput, we generate high complexity data, and it's quite expensive step. While ideal assay for biomarker verification, validation, and the FDA-approved assay should be a fast, high throughput, high sensitive and specific, and less expensive. The golden standard for such assays has always been ELISA immunoassays, but what we do in our lab, we try to replace ELISA with mass spectrometry, at least for a verification step in our pipeline. And we believe that mass spectrometry may provide additional advantages, such as multiplexing, and also verification of proteins for which antibodies do not exist at this time. When we talk about proteomics and biological mass spectrometry, we have to first consider the challenge of proteomics. And this challenge stems from the fact that the range of protein concentrations in the blood plasma is 11 orders of magnitude. It's really an astronomical range of magnitude. So proteins which we can measure and quantify by mass spectrometry in biological fluids is only a fraction of proteins identified by mass spectrometry, and again, it's only just a fraction of proteins present in biological fluids. So we can reliably quantify high and medium abundance proteins in biological samples, but it turns out that most interesting proteins and clinical biomarkers are low abundance proteins in blood, so they may not be amenable to quantification or to robust quantification and even identification by mass spectrometry. That's why we may need to use some additional approaches, some sample purification, protein enrichment. Another way around identification low abundance proteins is to use different types of biological samples. Even though blood plasma is an almost ideal fluid from the patient's and clinician's perspective, but it's the most challenging from the proteomics perspective. While other proximal fluids, such as saliva, urine, ascites, seminal plasma, amniotic fluid, may be enriched with biomarkers, especially with tissue-specific biomarkers, so it may be much easier to identify and quantify and verify novel biomarkers in proximal fluid relative to blood plasma. In this webinar, I will not present many details on shotgun and bottom-up proteomics, which is a very standard approach that we use in our lab, but I will say a few words about quantitative mass spectrometry by targeted proteomics or selected reaction monitoring. These are type of quantitative assays which we heavily use for biomarker verification. And this type of assays, for each protein we select a unique peptide, and for this peptide we monitor in the mass spectrometer several fragments. So this type of assays are done in the triple quadrupole mass spectrometer. So our peptides are first separated by liquid chromatography, ionized by electrospray ionization. Peptides are filtered in the first quadrupole, fragmented in the second quadrupole, and fragments are separated in the third quadrupole. Intensities of fragments are integrated, and the area under the curve is used to quantify the abundance and even absolute concentration of each protein in biological samples. And these days up to 500 proteins can be multiplexed in a single SRM assay. When it comes to development of SRM assays, we need to select prototypic peptides specific for our protein of interest and also select specific transitions. For this purpose, we use our in-house identification data, but also some public databases which accumulated lots of information and are very useful for development of this type of assays. This is Peptide Atlas and also GPM databases. And to select specific transitions, we also use our experimental data, some data in Peptide Atlas, and in silico prediction as well, using some software packages such as Pinpoint and Skyline. Since sample preparation is a bottleneck of any proteomic experiment, we do all the steps of proteomic sample preparation on 96-volt plates to be as much reproducible as possible. So all steps of protein denaturation, alkylation, digestion, and extraction are done on the plates, and then peptides are separated by nanoliquid chromatography, ionized by nanoelectrospray ionization, and the triple quadrupole is used to analyze our peptides. In the next few slides, I will talk about a specific example, which is a discovery of biomarkers of urological disorders. In general, urological disorders present a set of pathologies such as prostate cancer, testicular cancer, male infertility, which affect several glands in the male reproductive system. And the major point is that non-invasive differential diagnosis remains an unmet need for most urological disorders. So new biomarkers are needed, and this is a major goal of our work to develop these new diagnostics. In our work to find biomarkers of urological disorders, we focus on the proteome of seminal plasma. Seminal plasma is a proximal fluid which contains proteins secreted by 5 major tissues and glands: testis, epididymis, corpus gland, seminal vesicle, and prostate. Another unique advantage of seminal plasma is that it contains many tissue-specific proteins, and actually the number, for example, testis-specific proteins exceeds the number of tissue-specific proteins in any other human tissues. And this may be, again, an advantage to develop a very specific assays which reflect a pathology in the specific gland or tissue in the male reproductive system. In the last 5 years or so, we identify more than 3,000 proteins in seminal plasma samples from healthy men and also men with prostate cancer, prostatitis, and several categories of male infertility. And now we use this proteome as a foundation to develop quantitative assay, come up with some promising biomarker candidates, and to verify these candidates in seminal plasma. One of the most successful projects so far was the development of discovery and identification of markers of azoospermia, which is one of the medical conditions of male infertility. Azoospermia is characterized by non-measurable level of sperm in the semen, and it has two forms. Non-obstructive form, which is a result of impaired maturation of germ cells in testes, and obstructive azoospermia, which is caused by physical obstruction of vas deferens. And, for example, vasectomy can be a surgical obstruction. There is a clear unmet need to discover new biomarkers for non-invasive diagnosis and for differentiation between obstructive and non-obstructive forms, and can be useful in the clinic to replace diagnostic testicular biopsy, which is currently the only method to use in the clinic to differentiate between these two forms, obstructive and non-obstructive azoospermia. When we started this work on discovery of biomarkers of azoospermia, we had a hypothesis that testis-specific proteins should be absent in seminal plasma of obstructive and post-vasectomy patients simply due to physical obstruction, and we thought that some of these proteins may be also decreased in non-obstructive azoospermia. We developed a very well phased biomarker discovery pipeline, and we started with protein identification by mass spectrometry, then candidate qualification, SRM assay development, verification and validation, and our goal was to go through all these thousands of proteins and come up with a number of few proteins which can be useful in the clinic. So today I will just present some of the most interesting points of that work on biomarker verification and pre-validation. For biomarker quantification and verification, we use SRM assays, as I previously mentioned. In this specific case, these were SRM assays for 20 proteins, and we also used stable isotope dilution, which means we had heavy isotope labeled internal standards for each prototypic peptide. We worked with a direct digest of seminal plasma, so there was no enrichment of seminal plasma, which increased reproducibility of assay. And this assay allowed us to measure accurate relative abundances or even absolute concentrations of proteins if we consider complete trypsin digestion. And the limits of detection of our assays varied from 10 to 200 nanograms per mil. So using this assay, we proceed to biomarker verification and pre-validation. On this slide, I presented our verification data for 18 most promising proteins in 119 seminal plasma samples. These were samples from 3 different groups: normal seminal plasma samples, seminal plasma from non-obstructive and obstructive and post-vasectomy patients. Using SRM assays, we generated lots of data for all these proteins, and each point at this plot is a concentration of a specific protein in the specific group of samples. So when we completed this verification, we realized that some of the proteins perform better, some perform worse, but actually we were happy to see that testis-specific proteins outperformed as the most promising candidates. At that point, we saw some proteins such as taxon 1, LDHC, and ECM1 that differentiated between these 3 different groups with actually very high specificity and sensitivity. We also had 2 proteins which used as control proteins, and these prostate-specific proteins were not differentially expressed in any of the infertility conditions, which makes sense. At that time, we also realized that if we combine two different proteins, we can differentiate between three different conditions or three groups of samples. For example, if we combine taxon 1 and ECM1 protein, we can differentiate between normal, non-obstructive, and obstructive forms. Taxon 1 protein was found to be highly abundant in normal samples, while actually below the limit of detection of our assay in non-obstructive and obstructive form, while ECM1 protein was abundant in normal and non-obstructive form and downregulated in obstructive or post-vasectomy samples. So what was interesting, that these markers could differentiate between these conditions with very high sensitivities and specificities close to 100%, which was also a very rare case for biomarker performance. So one of the proteins, ECM1 protein, there was a reliable ELISA assay available, and we use this assay to verify our mass spectrometry and the proteomic findings, and so we confirmed them by ELISA assay, and we also established a preliminary cutoff level for this protein in seminal plasma at 2.3 micrograms per ml. For another protein, taxon 1, there was not much data available, not much literature and experimental data, so we decided to look in more details in the possible functional role of this protein in azoospermia. As the first assay, we used the immunohistochemistry staining of taxon 1 protein in testicular tissue, and it turned out that taxon 1 is a membrane protein. It's expressed exclusively in germ cells at the level of spermatocytes, but it's not expressed in Sertoli cells, Leydig cells, or any other type of cells in the testicular tissue. So this protein was very specific and cell-specific in the testicular tissue. Then later we used the human protein atlas to look into the tissue-specific expression of this protein in all human tissues, and it turned out that this is a very tissue-specific protein. Basically, it's a protein with a ultimate tissue specificity. It turned out that it's expressed in only one of 76 human tissues and in one cell type. So this makes sense why this protein may emerge as a very specific biomarker of azoospermia, because it's expressed only by one cell type, and the cell type is actually responsible for the development and the progression of the male infertility. We also decided to look in more details in the expression of this protein in a non-obstructive azoospermia. I just want to remind that using our mass spectrometry, we could see this protein at a very high abundance in normal seminal plasma, while their levels were actually below the limit of detection of our SRM assay in non-obstructive and obstructive azoospermia. This was not very surprising for obstructive and post-vasectomy form, while it was a bit of surprising for non-obstructive azoospermia. So non-obstructive azoospermia consists of three different subtypes. It's hyperspermatogenesis, maturation arrest, and Sertoli cell-only syndrome, and these three subtypes are actually clinically different. For example, in the Sertoli cell-only syndrome, germ cells are absent in the testes, and men with Sertoli cell-only syndrome are usually infertile. So we decided to use immunohistochemistry profiling to look at the taxon 1 expression in the testicular tissues from patients with different diagnoses. We found that taxon 1 was expressed in very high levels in tissues with normal spermatogenesis. There was no staining whatsoever in tissues with Sertoli cell-only syndrome, and there was some expression of this protein in hyperspermatogenesis and maturation arrest. So there was some discrepancy between immunohistochemistry and SRM data, so we could see the expression of these proteins in testicular tissues, while we did not see this protein in the seminal plasma. So we decided to develop a more sensitive assay based on immunoaffinity enrichment of taxon 1 protein followed by SRM detection in the mass spectrometer. We used the polyclonal antibody to enrich taxon 1 protein, and this assay was much more sensitive than just SRM assays. So its estimated limit of detection was around 5 nanograms per ml. And with this more sensitive assay, now we could see a taxon 1 protein in some of the non-obstructive azoospermia samples with a hyperspermatogenesis and maturation arrest subtypes, but there was no protein in the Sertoli cell only or obstructive and post-vasectomy seminal plasma samples. When we combined all our data for taxon 1 expression in the seminal plasma and the tissues, we proposed a model of expression and secretion of this protein in the seminal plasma. In the normal spermatogenesis, taxon 1 is expressed in the tissue. Germ cells mature into spermatozoa. Spermatozoa move into epididymis, and the taxon 1 is cleaved inside epididymis by a very specific enzymatic mechanism, and as a result we see a very high concentration of this protein in seminal plasma. In hyperspermatogenesis and maturation arrest, taxon 1 protein is expressed, but actually germ cells do not mature into spermatozoa, so they never move to epididymis, so there is no specific cleavage by this enzymatic mechanism in testis. So the level of taxon 1 that we see in seminal plasma is probably a result of some non-specific cleavage or shedding this protein from germ cells. And in the last subtype, Sertoli cell only syndrome, germ cells are absent in testis, so taxon 1 is not expressed, and the level of this protein is less than 5 milligrams per ml in seminal plasma, or even theoretically 0. Our data also allowed us to propose a potential method to distinguish between these two forms of obstructive and non-obstructive azoospermia. Using our two-marker analysis, we can differentiate not only between obstructive and non-obstructive form, but also rule out Sertoli cell only syndrome from hyperspermatogenesis and maturation arrest. And this is actually very important from the clinical perspective. It turns out that men with Sertoli cell only syndrome have a very low chance of sperm retrieval by testicular sperm extraction, which means a very low chance for in vitro fertilization. So testicular sperm extraction, which is a full-scale surgery, can be avoided for these men, and this may be very important. This assay may be very important from the clinician's perspective. Currently, we are working on development of immunoassay for taxon 1 protein, and so we have capabilities in our lab to clone and express this protein and also develop antibodies. So we already cloned this protein in yeast, produce recombinant proteins, immunized mice, and produce some good monoclonal antibodies, and we're currently optimizing our ELISA assay. And as soon as we have an optimized ELISA assay, we go for preclinical studies. And finally, I just want to mention that we also work on the prostate cancer biomarkers in seminal plasma. And we focus on two unmet needs in prostate cancer diagnosis. First need is to identify markers that complement serum PSA test and increase specificity of detecting cancer. And second goal is to identify markers that discriminate between indolent and aggressive cancer. We already identified a list of most promising candidate proteins in seminal plasma, and currently we are at the stage of verification of these candidates in the independent set of samples, and hopefully at some point we'll be able to present new data on prostate cancer biomarkers in seminal plasma. Finally, I would like to summarize our presentation. I want to mention that the clinical use of each biomarker should be defined well in advance, so we need to know the clinical use of the novel biomarker well in advance. We believe that it's important to have a clear criteria for selection of biomarker candidates. In our case, we rely on secreted proteins and also on tissue-specific proteins. We believe there is a lot of future, especially in tissue-specific proteins. We also use the proximal fluids to facilitate biomarker discovery, and also some of these proximal fluids may be used for final non-invasive diagnostics. We also believe that SRM assays could close the gap between candidate identification and biomarker verification, and this was a gap in the biomarker discovery pipeline which existed for many years until introduction of SRM assays. And we hope that upon rigorous validation, our candidates Taxon 1 and ECM1 could possibly emerge as very sensitive and specific male infertility biomarkers. Finally, I would like to acknowledge people who are involved in this project and working at Mount Sinai Hospital at this project. And finally, I would like to thank you for your attention.
Thank you for your presentation, Eleftherios and Andrei. Both speakers are now on hand to answer any questions you may have. To ask a question, please type it in the questions box and we will answer as many as we can live today, and we will follow up by email for those we don't have time for. So our first question, and I think this one is directed to Eleftherios. A lot of people are talking about the PSA biomarker for prostate cancer. Is this a good biomarker or a bad biomarker?
Yes, PSA has some great advantages and some big disadvantages. The main advantage of this marker is that by using it, you can detect more than 90% of prostate cancers with a very simple and non-invasive technique. Unfortunately, though, prostate cancer comes with a number of different flavors. It's a heterogeneous disease. Some of them progress very quickly, and these are the ones that you want to diagnose. Some of them progress very slowly, and these are the ones that you do not want to diagnose, because if you diagnose, it's called over-diagnosis, followed by over-treatment. So unfortunately, the PSA test identifies both the so-called clinically relevant, clinically important prostate cancers and clinically indolent prostate cancers. Another major disadvantage of PSA is that it is also elevated in benign prostatic diseases such as benign prostatic hyperplasia and prostatitis. So to summarize, a PSA allows you to detect a lot of cancers that you want to detect, but unfortunately it also detects cancers that you do not want to detect because they are indolent, and it's also elevated in benign diseases, and then getting many people to do prostatic biopsies, which of course we do not want to do.
Excellent, thank you. And our next question is directed towards Andrei, and they were wondering how many peptides were selected for each protein, and also how do you select which peptide to represent the protein?
I think it's a very good question. In our final assay, we measured one peptide per protein, but during method development, we considered multiple peptides, and we selected the one which was reproducibly detected and had a high sensitivity. We also search peptides through some proteomic databases to make sure the peptide is unique and therefore represents only our protein of interest and no any other protein.
Excellent. And just sort of following on from that, are the LOD values in your talk, are they for peptides or are they for protein levels?
Here the LODs are at the peptide level because we're actually measuring peptides, and we are using peptides as internal standard. So yes, they are at the peptide level.
Excellent. Thanks, Andrei. And the next question goes to Eleftherios, and it asks, if a marker is 90% sensitive and 90% specific, does this mean that it is good for clinical practice?
Yes, although the numbers look pretty good, unfortunately, the sensitivity and specificity cannot determine if a marker could go to clinical practice. The sensitivity specificity of the marker has to be balanced against a clinical question. For example, if your purpose is to diagnose as many cancers as possible with relatively low specificity, which means that a lot of people will be false positives, if you have a confirmatory test that is relatively easy and non-invasive, then the procedure may be acceptable. And the classical example of this is PSA again, which, as I mentioned before, has excellent sensitivity but poor specificity, and yet it is used because the biopsy can determine who has and who doesn't have prostate cancer. On the other hand, it also depends how you're going to use the marker. If you use it in a screening scenario, in which case you test asymptomatic populations, and the disease is relatively rare, like ovarian cancer, then you may end up even with 90% specificity with a lot of false positives. So just to summarize then, if you want to detect in a screening scenario as many cancers as possible, high sensitivity is of course desirable, over 90%. The specificity requirement, it also depends on the prevalence of the disease. It is very well known that if you screen for a rare disease, you need to screen a lot of people to get one true positive. And if your sensitivity, your specificity is only 90%, you will end up having a lot of false positives. For example, if you screen 1,000 people, you can get one person that has, let's say, ovarian cancer, and at 90% specificity, you're going to get 100 people who are false positives. So you can understand that in this kind of scenario, you get 101 positives, of which one is a true positive, and the other 100 are false positives. And delineating who is a false positive or true positive, it may require invasive procedures like laparotomy in the case of ovarian cancer. So just to summarize, then depends on the clinical scenario on what is the acceptable sensitivity specificity for the biomarker.
Excellent. And I think this next question is directed to Andrei, but please feel free to step in if it's not Eleftherios. But the question asks, was there any glycosylation variation of the protein that you identified during the proteomic process? And are there any useful exploitations to detect the glycans on a protein as a biomarker?
I think this specific case, we were not looking for any glycoforms. So the peptide we measured, they did not have any glycan post-translational modifications. And typically we don't look for glycans on proteins as a biomarker, but I think there was one of the works on the kallikrein 6. Maybe you can add.
Yes, Eleftherios, here, as Andrei said, we discovered one ovarian cancer biomarker, kallikrein 6, which we have identified major differences in the glycosylation of the marker that is produced by cancer cells versus the marker that is produced by normal cells. We found that the cancer cells produce kallikrein 6, which is loaded with sialic acid, as opposed to not much sialylation in the KLK6 that comes from normal tissues. The difficulty, though, in establishing this kind of differences, which definitely exist between cancer and non-cancer, in the practical situation is not as easy, because if there is a difference in the glycation, you have to detect it with a mass spectrometry technique in a complex mixture like serum. And we found it very difficult to detect in serum these glycosylation differences between the KLK6 isoforms. So just to summarize again, the glycosylation differences exist. There are biomarkers based on glycation differences, but they are relatively difficult to quantify in biological fluids, especially when present at very low concentrations.
Excellent. And the next question goes to Andre. And during the presentation, I think you mentioned that you were able to measure 500 proteins in a single SRM assay. How long was the runtime? Was it quite rapid?
Unfortunately not, because if you want to multiplex hundreds of proteins, we need to have long gradients. So it's around 1 hour chromatography gradients. So at this point, these type of assays are not very quick. So I would agree that with these long assays, you probably cannot go to the clinic. While in the clinic, you need a very rapid assay and probably a possibility to screen as many as thousands of samples per week. I think that's one of the requirements of clinical assays. And seems like SRM assays are not there yet if we talk about hundreds of proteins, but we hope that at some point it may be there.
Excellent, thank you. And here's the next question. Are there more robust assays than ELISA to screen those candidate biomarkers?
More robust assays?
Yeah, than ELISA to screen any sort of candidates.
Yeah, we are personally, Eleftherios, I'm not aware that there are methods that are more robust than ELISA. As you know, the ELISA methodology is now well established for over 30 years, and good ELISAs have outstanding sensitivity in the level of 1 picogram per milliliter. You can go down with precision between 3 to 5%. So these techniques, and the specificity of course depends on the antibodies, but they could be very specific assays. So it's very difficult to compete with those, but obviously any other analytical technique, fluorometric methods, methods based on mass spectrometry, again depending on the abundance, they could be employed, but I think ELISA is probably unraveled if we consider the sensitivity, specificity, and convenience, speed that you can get with.
Excellent, thanks for that. And I think this question is directed to Andre, and it's going back to the LOD again. And they were wondering, how do you go about measuring the LOD of a peptide that is based on a sort of synthetic peptide?
Well, here it's very simple. We do it actually in a reverse order. We have dilution series of synthetic peptides spike into biological fluid of the same amount of biological fluid, and then we just calculate the ratio, and we have a calibration curve, and we believe that heavy to light will be the same ratio as light to heavy, which actually should be the case. So that's the way that we measure LODs. Brilliant.
Thanks for that. And here's the next question. Can mass spec proteomics identify key candidates missed out by gene microarrays and sort of a bit of a tongue twister here, suppression subtractive hybridization?
Can you repeat the question, please?
Yes, sorry about that. So can mass spec proteomics identify key candidates that would otherwise be missed out by either gene microarrays or suppression subtractive hybridization method?
Yes, a version of the subtractive hybridization technology that has been used in genomics some time ago is the idea of delineating proteomes in a quantitative fashion of fluids that one represents normal situation, the other one represents cancer. I'll give you an example. We have delineated in the recent past the proteome of pancreatic juice from normal individuals or pancreatic juice from patients with pancreatic cancer. If you do this in a semi-quantitative fashion, like with spectral counting on extracted ion current, you can compare the intensities, presence, absence, or even quantitative comparisons between all the proteins in the normal, so-called physiological, and cancerous pancreatic juice. And with this kind of approach, a subtractive approach, we were able to identify unique proteins that are present only in the pancreatic juice from cancer patients. So yes, you can use this kind of comparative approach to identify molecules which are present in one condition, for example cancer, versus another condition, example normal. Yes.
Excellent. Thanks for that, Elatharios. And the next question goes to Andre, and it asks, what are the biological functions of ECM1 and TEX101 proteins? Are these proteins crucial for reproduction?
When we look at literature to learn about biological functions, it turned out that ECM1 protein is actually extracellular matrix protein, and I think it's involved in just, it provides some media characteristics of the seminal plasma, but it's probably not directly involved in infertility, while TEX101 protein turned out to be a much more interesting protein. And we found in the literature that knockout mice were infertile, or the knockout TEX101 mice. And this seems to be a very interesting protein with an ultimate tissue specificity. And as we see, it can be a great biomarker of male infertility. And this protein may emerge as an interesting target in the male reproductive studies.
Excellent, thanks for that. And the next question is also for you, Andre. What are the potential limitations of mass spectrometry-based protein assays for use in the sort of clinical laboratory?
When it comes to use of mass spec in the clinical laboratory, these limitations may be, first of all, sensitivity. So mass spectrometry assays are not very sensitive yet. Then this can be reproducibility. We need a much better reproducibility of protein assays. The throughput, I think it's also, we're liking on throughput. Again, with a good assay, we should be able to analyze thousands of samples in a week. So we're still missing this. And I think finally it's probably standardization. So we need to make sure that the results provided by different laboratories are standardized. So we need to develop protein standards for this purpose. And I guess as soon as we work on these issues, sensitivity, reproducibility, throughput, standardization, mass spec may emerge as a great tool for clinical laboratory analysis.
Excellent. Well, I'm afraid that's all we have time for. I'd like to thank our speakers, Eleftherios Diamandis and Andrei Drabovic, for their presentations and for answering the questions. I would also like to thank the webcast sponsors, Thermo Scientific, and of course you, the audience, for watching. Remember, if you have a question, and we'll try and get back to those that we didn't have time for just now. You can watch this webcast again at any time on demand at nature.com/webcasts. If you're watching on demand and have a question, please type them in up to a week after the live broadcast and we'll get back to you by email. Thanks for watching.